Well, thank you, Jesse, and thank you for letting me present our research on nitrogen management in viticulture and enology. We have a project with a number of different objectives that were fairly diverse. So we were interested in looking at soil and foliar nitrogen applications and its impact on fruit quality. We wanted to also see whether these treatments would alleviate leaf roll virus symptoms. And we wanted to also evaluate whether we could use aerial imaging with these trials for precision management in the vineyards. We also looked on the wine side to see if these treatments had impacts on wine quality and also the influence of amino acids and diammonium phosphate additions on wine quality. So we looked at Cabernet Sauvignon and Pinot Gris, which I'll focus on here, but we also looked at Cab Franc and Sauvignon Blanc. And this is just a standard way we've set these trials up. We used just the standard management practice in the vineyards, so whatever the vineyard was applying. And in this case, about 40 pounds of nitrogen per acre. And then on top of that, the two treatments, we added 20% more nitrogen or 40% more nitrogen. This was all through fertigation. Or we applied foliar sprays on top of the standard management practice. So we either applied 2 or 3 foliar state sprays, and this was 2% urea. And we applied those at about 10-degree Brix, and so this is just prior to veraison. And using Brix is a really good way to consistently apply rather than just trying to estimate when veraison is happening, which is usually at the target area for urea sprays. You can see here just the general schedule that we would use, say in the Cab Sauv. The sprays were separated by about 2 or 3 weeks, and it depended on the variety. So shorter season varieties, you're around 2 weeks between applications, or something like some of the red varieties were more like 3 weeks between application. So what we found was with the urea sprays, we were able to increase yeast assimilable nitrogen by about 2-fold. And the increase was due to incorporation of amino acids. So it increased the amino acid content. And we didn't find any increases in vine vigor or yield. And this is an important point because early nitrogen applications can often cause increases in vine vigor and affect yield and quality. Vegetative aromas are common when you apply nitrogen early on in the season. And so these foliar sprays were more targeted later in the season to avoid that reduced quality in terms of vegetative aromas. So you can see here with these foliar sprays, we can increase yeast assimilable nitrogen by about 2-fold. And this is in the range that we wanted for fermentation. So we're able to increase it enough using these sprays to either reduce or eliminate diammonium phosphate in the winery when we make wine. So this is a very, very positive result that we had. What we did find was, and this is also in the literature, there's a bit of confusion in the literature whether these work or not. And what we found is white varieties seem to be a little more receptive to this treatment than red varieties. So when we looked at leaf roll virus to see if nitrogen would alleviate some of the symptoms, we found that there was no effect. What our thinking was is that nitrogen would produce more chlorophyll, then there would be more energy produced in the plant, and then that would maybe advance maturation. And that we didn't find that at all. We used pairs of infected, non-infected vines. We had about 40 pairs that we would look at and treat them with these nitrogen treatments. And we found out that this wasn't a good approach for trying to control some of these quality issues with leaf roll. So another thing we did was look at nitrogen in the vineyard and try to use aerial imaging in order to model nitrogen status for precision management. So we used our trials We used some imaging techniques, and really we wanted to be able to predict areas in the vineyard of low or high nitrogen for management purposes. So this is a very rapid way that you can estimate your nitrogen status. So we were successful in doing this, and we used a special approach to this that Brad Estegard applied for us. But I'll just go through the typical way aerial imaging is done is through this technique called nadir, which is a top-down approach. It's two-dimensional. Basically, you get an image of the tops of vines here. And so this is our trial. All the green dots are where our experimental plants are. This is just expanded here. So each plant there we get, so this is about a 1 meter diameter circle. We get about 100 points per vine using this technique, so 100 data points. And then we can average those into a single data point for each vine in order to use for modeling. So this is a typical NDVI picture, and this is something that you might see commercially. So what we did was looked at a different approach called multispectral point cloud. And what this is we get a lot more data. So each data point, there's about 1,000 to 4,000 data points as opposed to 100. So it's about 10 to 40 times more data points per vine. We also, for each data point, we can collect 3D information, so the spatial information, so X, Y, Z coordinates. So we know exactly where in the vine each point is. And with each point, we can also collect up to 10 different wavelengths in the visible and near infrared spectrum. Not only that, we can also take a look at the sides of the vines instead of just the top. So now we're getting a lot more information from the canopy rather than maybe 20% from the very top. We're actually able to measure the whole canopy on both sides. And you can see how many data points, so down in the blue here, each one of these little ones is a data point with all that information. So we can get much more accurate in terms of the averages we get per vine for modeling. And what we found when we use this system, these are all different indices. So you can see NDVI, which most people probably know at the top, but these are all different types of indices. And when we use the nadir, so the two-dimensional top-down approach, these are the indices in yellow here that were significant and correlated well with our ground data. When we use this point cloud system, you can see there's a lot more indices that we can use. So that enables us to get much more accurate models. And we were able to, with 90% accuracy, predict what nitrogen levels were in the vineyard. So on the bottom here, this is what we actually measured in the vineyard, and this is what we predicted through our modeling. So 90% is very good for these types of models. So we're able to use this to, say, fly over a vineyard in general and predict whether areas are going to be low or high nitrogen, and that will allow easier precision management within the vineyard. So we also looked at the impact of foliar nitrogen on wine quality. With the foliar nitrogen, we saw that we could increase yeast assimilable nitrogen by basically double. And this was sufficient for fermentation where we didn't have to apply diammonium phosphate in the winery for fermentation. This is really important. So the nitrogen that was sprayed on the leaves was taken up into the plant and converted into amino acids. And this creates an environment for fermentation, which is much more diverse than just adding diammonium phosphate. So it's important to, when you can, get the nitrogen into the fruit in the field rather than just applying it in the winery, because really it's unidimensional when you have diammonium phosphate too much, and it doesn't really provide a lot of different flavors and aromas, whereas amino acids do. So we measured amino acids, and each one of these amino acids here provide different types of flavor, aroma, mouthfeel, texture, and also protein stabilities. And each one has their different properties. And I'm sorry the graph is so small, but can't go into detail about each one. But if you look over here, these are the totals. And when we have the blue and red, which are the foliar nitrogen applications, we can see total amounts of amino acids are about double. So this is extremely important for winemaking because when the yeast uses these amino acids for a nitrogen source, they break off portions that are flavor and aroma compounds, which are extremely important for producing wines that have much more depth in terms of their characteristics. We looked at the influence of amino acids on quality. I'm just going to show you one example of our some sensory evaluation we did on Sauvignon Blanc from these treatments. And if we compare, so citrus flavor is in the blue, tree fruit flavor, and tropical aroma. So if you compare the colors across here, these are the treatments in the bottom, and the flavor intensity, we can see that there are some differences between these treatments in terms of the sensory quality of the wine. So this isn't to say that people should be doing it one way or the other. It's just to say that you can change the wine style and the quality and types of wines that are made by incorporating nitrogen into the fruit later in the season. And by applying foliar nitrogen later in the season, you can avoid some of those negative impacts of high nitrogen early in the season, which creates vigor in the vines, can change crop load, and can increase vegetative flavor and aroma, particularly in the form of pyrazines. So just to summarize, we can increase YAN with foliar nitrogen by about 100%. And this is basically the more sprays you apply, the more YAN that's going into the fruit. And this is in nitrogen deprived production. So we didn't test this on very high nitrogen areas, which we don't have a lot of in the valley here. Typically in the south, we're looking at fairly low nitrogen levels in the vines and fruit. So vine vigor not impacted by foliar N. We weren't able to alleviate leaf roll symptoms with these treatments. The yeast assimilable nitrogen is mostly in the form of amino acids when we apply these foliar treatments. And the higher amino acid content, I mean, this has been shown over and over. It really contributes to complexity of wines and changes that sensory profile. And then using some advanced techniques in spectral imaging, we were able to successfully predict in the vineyard where areas of high and low nitrogen is. And I'd just like to thank everyone. AAFC, we have lots of collaborators on this. Collaborators at Covey, UVic, and our technical support, many co-op students, and of course industry and financial support through the Wine Grape Council, AAFC, Arterra, Andrew Peller, and CGCN. Thank you.